Hi everybody, this is your lecture on enzymes, which are our helper protein molecules. And on this first page, you can see an enzyme here in the blue, as well as this purple enzyme below. So those are two different images of what enzymes look like. So fill in that unit title, enzymes, helper protein molecules. So life is built on chemical reactions. We have the sun, giraffes. So plants use the sun to make energy, and giraffes eat plants to make energy. And then lions eat those giraffes to make energy. It's a kind of sad picture. So chemical reactions of life. Oh, you can write all that stuff down about the lions and the giraffes and the plants. Are necessary in processes of life. Chemical reactions of life are necessary in the processes of life. So in different processes, we need to build molecules, which would be synthesis. So building molecules is called synthesis. And when we take two things and we put them together into one molecule. Or you can have breaking down of molecules, which is called digestion, which is illustrated here. Nothing works without enzymes. So how important are enzymes really? Every chemical reaction in a living organism requires enzymes to work. So they are extremely important. They're required for building molecules. So we have that same picture from before, and again, that's called synthesis. So we would use synthesis enzymes to do that. And they're required for breaking down molecules. So you would use digestive enzymes to do that, since breaking down molecules is called digestion. When we write that we have this, we would have enzymes in chemical reactions. We show the substrates and we show the products, and then the enzyme goes right over the arrow to show that it's helping out with that chemical reaction. And enzymes speed up reactions, and because they speed up reactions, they're called catalysts. Some examples of synthesis enzymes. You can write this on the right-hand side under where it says examples. You can draw this picture. So we have the enzyme that comes in, and it puts those two molecules together. It synthesizes a bigger molecule. Or you can have digestion. And in that case, the enzyme is going to come in, and it's going to break those two molecules apart and make two new molecules that are smaller. So again, you can draw those pictures under examples on your right-hand side. Enzymes are proteins. Each enzyme is a specific helper to a specific reaction. And each enzyme needs to be the right shape for the job. Shape is so important when it comes to enzymes, and any protein really. Enzymes are named for the reaction they help. So one enzyme only does one job because of its shape. So sucrase breaks down sucrose. You can write this in the examples column. Proteases break down proteins. Lipases break down lipids. And DNA polymerase builds DNA. So the names are going to tell you exactly what it does. And see this ASE at the end of these words? ASE, that shows us it's an enzyme. Enzymes are not used up by the reaction. They are not changed by the reaction. So they are able to be used over and over again. They're used only temporarily, and they're reused again for the same reaction with other molecules. But you only need a little bit of enzyme to help in many of the reactions, so that's a really good thing. So here's our picture, and I want you to draw this on the right-hand column on the next page. We have our substrate, which is this yellow molecule that's coming in. This is the molecule that the enzyme is going to work on. And we have our purple enzyme. So the substrate is going to go into the purple enzyme. And in this case, we have a digestion enzyme. And the substrate is going to go into the enzyme at the active site. So the active site is where the substrate fits into the enzyme. And a product or products are going to be created at the end. And you'll notice that we still have the enzyme here. It did not get used up. It's the shape that matters, and they call this a lock and key model. The shape of the protein allows the enzyme and substrate to fit. So this is like the lock. The enzyme is like the lock, and the substrate is like the key that fits into it. So there's a specific enzyme for each specific reaction because only one key is going to fit in any lock. So once again, we have the substrate. The substrate goes into the active site here. The enzyme is going to break down the substrate into two separate products. The substrate will leave, and we have our enzyme ready to go for another molecule. So we have 1, our active site is available for another molecule of the substrate. 2, the substrate has entered the enzyme at the active site. And 3, the substrate is converted to products. So you can write here, substrate binds to enzyme, and substrate is converted to products. Those are the steps. Now here's some enzyme vocabulary. An enzyme is a helper protein molecule, meaning that it helps chemical reactions occur in the body. A substrate is a molecule that enzymes work on. Products are what the enzyme helps produce from the reaction. So the substrate is the molecule coming in to the enzyme at the beginning. The products are what's coming out at the end. And your active site is the part of the enzyme that the substrate molecule fits into. So different things are going to affect enzyme action. The first thing is that you need to have the correct protein structure. So you have to have the correct order of amino acids. So if you look to this picture on the right-hand side, you see that each of these little bubbles is a different amino acid. Okay? And all of those amino acids are going to create the protein. If even one of them is wrong, it could make a huge mistake in the protein's shape. And as I said before, shape is really important. Okay, so if you flip, we have temperature, because the enzyme has to be the right shape. That's also going to affect enzyme action, as well as pH. And why? The enzyme has to be the right shape. So the things that affect enzyme action affect it because they affect the shape of the enzyme. That's very important. So what creates the order of the amino acids? And the answer is DNA. DNA determines the order of the amino acids in the protein, which determines the shape of the protein. And this one is the right shape, so that's good. However, if we have a mistake in the DNA, we may get a different chain of amino acids. So we can see that here we have 4 reds in a row, and here we have 2, a blue, and another red. So that's gotten messed up. So, because they're not in the right order, the protein is going to be in the wrong shape, which is bad. Because as we heard before, shape is very important. Otherwise, that substrate is not going to fit into the active site. Okay, so temperature. Temperature affects the rate of enzyme activity. There is an optimum temperature for enzyme activity. And in that optimum temperature, that has the greatest number of collisions between the enzyme and the substrate. Because remember, as temperature increases, molecules are going to move faster, so they're going to hit the enzyme more. Human enzymes, the temperature, not surprisingly, is between 35 and 40 degrees Celsius, because that's the same as human body temperature. If you raise the temperature too high, it will denature the protein, which means the protein unfolds or loses its shape. If you lower the temperature too low, the molecules are going to move slower. So they're not going to go into the enzyme as much. They're just not going to hit it. So there'll be fewer collisions between the enzyme and the substrate. Okay, so we need to describe what's happening here on your note sheet. So we have our reaction rate, on the y-axis, and the x-axis has the temperature. Now we're talking about human enzymes here. So our ideal is going to be 37 degrees Celsius, which is body temperature. So enzymes that are in an environment that's too cold are not going to work very well. Their reaction rate is going to be very low. Enzymes that are in temperatures that are too hot are going to lose their shape, and their reaction rate is also going to be very low. So the perfect reaction temperature is this one right here. We have a very high reaction rate, meaning many substrate molecules are being broken down. So how do cold-blooded creatures do it? Okay, we have a bobcat, which is an endotherm like us, but snakes are ectotherms. So they don't have a specific temperature that they use. So their enzymes in their body, they may have multiple enzymes that do the same thing at different temperatures, or it might be an enzyme that works well in a larger temperature range. pH also affects the rate of enzyme activity. Changes in pH change the protein shape. So it's all about shape here. If I change the pH, the protein shape is going to change. Most human enzymes like a pH of between 6 and 8, so almost neutral, very close to neutral. Remember, neutral pH is 7. But that depends on where in the body they are. If you're in the stomach, which is pepsin, an enzyme in the stomach, likes a pH of 3 because your stomach is very acidic. So that enzyme works really well in an acidic environment. If you're in the small intestine, slightly basic, though that enzyme works best at the pH of 8. Okay, so we have neutral at 7. Most enzymes are going to work really well at neutral. Their reaction rates are going to be low if they're below 7, and low if they're above 7. Remember, the shape is just changing, so that's what's happening. Now, the stomach is about 2.5, 3, so pepsin, the enzyme in the stomach, is going to work really well right around 3. It's really adapted well for its environment. And then our intestines are slightly basic, right around 8. So the enzymes in the intestines work really well around 8. So again, they're very well adapted to their environment. So in conclusion, for enzymes, what matters? The shape. So please write down any questions that you have from today's lecture. You can write them on the back of your note sheet and bring them into class with you tomorrow. Thanks.